This is a neat paper that a mutual asked me to review a while back. It looks at the effects of tea and catechins on COVID, where catechins are natural chemicals that exist in food and medicinal plants, where they were able to show that the tea catechins, EGCG and TFDG, both strongly inactivated conventional COVID binding to the receptor binding domain of the spike protein, where Omicron subvariants were inactivated by green tea, matcha, and black tea, where some variants like BA1 showed strong suppressed infectivity, while this effect was less in other variants like BA2.75. So how they ran the study is they had healthy volunteers who consumed a green tea candy or a black tea candy, and then they basically spit in a vial and they infected those vials with COVID to see what would happen. So right off the bat, the big thing is that this is a static fluid. In our mouths, the turnover is constant, where you're always producing additional saliva as long as you're hydrated, where the virus suspension is then mixed in with that spit, and then after 10 seconds, the virus and tea mixtures were serially diluted, so different concentrations of liquid to virus. So for reference, the blue would be distilled water, light green is a sencha green tea, dark green is a matcha, brown is a black tea, and then olive or lime green is a bottled green tea beverage, where in general the matcha and black tea had very, very significant levels of decreased viral activity, viral concentration levels after the 10 seconds, and that bottled green tea beverages don't have the same effect as the others as a green tea, but they do reduce the viral concentration pretty significantly still. And like any good experiment, you should have a pre-control, so they have the spit prior to doing the saliva from the volunteers, and then they compared this to the treated with the mixed samples, and you see these decreases at higher concentrations, higher levels of decreases. And this one's really interesting. So they looked at the computational modeling of the spike proteins on these different variants, and then this would be the catechin of the green tea, that molecule in the green tea that would be blocking the receptor binding domain. So it would be blocking this area of that spike protein. This is a different version, so one's a stick model, one's a ball model. And if the virus is blocked from the tea, then it can't interact with a cell to cause that cell fusion. So what it does is it inactivates the virus by gumming up the receptor so that it can't do that. But there are other receptors on COVID. There's that N gene, the nucleocapsid protein as well. But at the very least, this is a very interesting thing. However, it's very important to note that this impact is temporary, where it will inactivate high levels of the virus very close to consumption, but as time progresses, even 5-15 minutes later, the inactivity of the virus is lost over time because your mouth is clearing itself out. So while this wouldn't be a great thing to use as like a prophylactic to prevent you from getting COVID or anything like that, what is incredibly interesting to me about this is, okay, so what if we aerosolized catechine molecules and inactivated COVID in the air by blocking those spike protein receptor binding domains? You could literally just like nebulize green tea like an air freshener, and that could be a way that we could work to sort of disable the virus while it's lingering in the air. And to me, that would be a very interesting study.